here's why we're here today. Citrus. What is citrus? This is the title and the authors of the paper in question. It's a pretty excellent algorithm at analyzing groups of data. So previously in Cytobank, we have Visney and Spade, and those are fantastic at finding heterogeneity inside of a sample. And Spade is great at comparing in a fold change manner between several samples. But really what Citrus does is allows you to leverage group-based statistical analysis to figure out what makes groups of samples different. So essentially, Citrus lets you compare groups of samples. Not only tells you what parameters makes those groups different, but how significant those differences are. So what are the applications? It's fantastic for biomarker discovery, for preclinical or clinical trials, for any reason you need to compare groups as opposed to individual samples. But what about the nitty-gritty? How does it go about doing this? Citrus begins by identifying clusters of phenotypically similar cells in all samples in a hierarchical, unsupervised manner. This is similar to how Spade produces its tree. Basically, it takes groups of phenotypically similar cells and puts them into these nodes. After that, however, Citrus uses regularized supervised learning algorithms to identify stratifying clusters and cell response features that are the best predictors of a known experimental endpoint of interest. So after it has built this phenotypic tree, it says, all right, now using these clusters, what's different between the different samples? So here's a brief little cartoon kind of describing what features in a group might look like. Group 1 is non-responders. Group 2 are responders to perhaps a compound in question. Group 1 has just one feature. Group 2 has a mixture, but feature 2 in this illustration is a stronger predictor of what might be happening between these two groups. So Citrus is a more complicated algorithm than, at least to set up, than Visney or Spade are. There's several different versions of the analysis you can run. First off, there's several different association models. So SAM is a correlative association model that has its roots in the analysis of microarrays. And you can run as many groups as you'd like through SAM. PAM is a predictive association model. And again, you can run as many groups as you'd like. And Lasso via GLMNet is predictive, just like PAM, but you can only run 2 groups through this analysis at once. However, you can run all 3 association models on your data, or the first 2 if you have many groups. So you can look at all the results that are generated. You're not stuck to just one of these per Citrus run. So in addition to the different association models you can use, you can also take a look at either medians for a particular Citrus run or abundances. So medians would be a great way to examine your groups if you have a functional marker. If you're looking at phosphosignaling, for example, you can use medians to say, what are the differences in these phosphosignaling markers between my 2 groups, and the median of a particular channel or set of channels. Abundances would be more appropriate if you have, for instance, a set of groups, and you're looking for the expansion of one population or the disappearance of another population. So it looks at how frequent the cells of interest are between those 2 groups. So regardless of what association model you use, you will get in your results a set of PDFs and a set of CSV files. Regardless, again, like I said, between whatever groups or whatever association models you use, you will get a marker plots PDF. And this essentially is the phenotype of the different branches in your Citrus tree. So this will use just the markers that you set as clustering channels in the analysis. And you can see here, there's a CD4 positive branch. Here you can see the CD33 positive node. However, even though this looks like a Spade tree, there's one important difference. From here, you can see that it looks like a series of conjoined nodes, just like a Spade tree would. However, these are hierarchical. So just like in a gating hierarchy, we have a series of nodes here. These events right here are contained in this node. And this node, all the events in this node are contained in this node. And it goes all the way back up. Might be tough from your computer screen to see, but these are actually arrows instead of just straight lines connecting these nodes. And these will kind of, if you follow them back, point you to the kind of originator node. And this is where all of the events are contained. So the most phenotypically pure groups or clusters of cells would be the ones at the end of these branches, which, as you can tell, this is why this node right here is a CD33 positive node. This node contains all the events in this one and in this one, so that's why it's colored kind of a yellow as opposed to a bright red for high CD33 expression. So similar to a spade tree in appearance, but definitely different meaning. This is the model error rate, and you get this graph when you run a PAM analysis on your data. Citrus PAM uses a few different features here, and this is a pretty complicated graph, but we'll dive into it. So you have a cross-validation error rate, a feature false discovery rate, and you have kind of 3 different versions of the results here. So Citrus with PAM uses cross-validation and false discovery rate to select the best model by choosing a parameter called the regularization threshold for that model, which is shown on the x-axis down here. This threshold controls how many features will be included in the model. The lower you are in the x-axis, the less features you have in the model. And the higher you go on the x-axis, the more features your model will include. So the cross-validation error rate comes from a process called cross-validation, where the data used to build the model are split into multiple training and testing sets to estimate the performance of the model. You want the cross-validation error rate to be low so the performance of the model is good. So one important thing to check on this graph is that the cross-validation error rate drops towards 0 as you increase the number of model features. If the cross-validation error rate doesn't get low as the regularization threshold goes up, then the model may not be working very well for your data. You might want to consider trying some different choices in your Citrus setup and to make sure the data your input is appropriate for Citrus. This is a good time to mention that Citrus obviously is a statistical, produces statistical results, and it's the more samples you use, the stronger those results will be. So you need at least 8 samples to have some confidence in the statistical results. Below 8, it gets a little shaky. You need at least minimum 3 samples per group. But again, 8 is much preferred, and above 8 is even better. So if you have maybe 2 samples per group, Citrus may not be the best tool for you, at least initially, until you can go back and repeat the experiment. So again, if the cross-validation error rate doesn't get low as the regularization threshold goes up, the model may not be working very well for your data, and you might want to consider trying some different choices. Citrus with PAM will give you 3 possible models to choose from. That's what these are. So these are little icons or indications about the models here. So CV-min and CV1SE are the models that have a good performance as determined by the cross-validation step and have the lowest number of features necessary to describe the difference between the 2 groups, or however many groups you chose to run. In contrast, CV-min and CV1SE are models. And there's the FDR constrained model. So this is this kind of yellow triangle down here. And you can see it's represented by this dot all the way on the other end of the graph. Citrus generates this by using a false discovery rate process to determine which features are statistically significant. This process requires a threshold, which is set at 1% by default. So you can see when this hops up a little bit to 1%, that's where this marker is. So when you see the blue line go above 1%, this is indicating the models above this threshold include features that are not statistically significant. And if you choose the FDR-constrained model, in contrast to the other 2 models, it'll include all the features that are significantly different between your groups, not just the minimum number of features, like these first 2 versions of the results include. So kind of the takeaway message here is to make sure that the cross-validation error rate drops towards 0 and gets as low as possible, and make sure that your models are below kind of this threshold of the feature false discovery rate. Another indication of a good run is that the CV-min and CV1SE models on this graph are not on this 0 axis here. And that means they don't need any number of features. A feature could be phosphosignaling, on one of your subsets, for instance. If there are 0 features required for the model, that basically means the algorithm thinks that it doesn't need any data to be able to predict which group a file should be in, and that's false. So this graph acts as a little bit of a kind of a quality check on how likely the run was to produce valid results. So again, this is just for PAM. This does not appear on the SAM version of the results. But in addition to the quality or the model error rate graph, you'll get several other PDF results. So the results of Citrus are not interactive SPADE tree like SPADE or another set of channels like Visney. It's a set of PDFs and CSV files that you can then examine for the kind of meaningful information. And this next set of results is what a lot of people will be looking for. So it's the feature plots PDF. Again, there's one for CV-min, there's a feature plots for CV1SC, and another feature plots for FDR constrained. So the model with the lowest estimated error rate, in this case, for this data set that we used from Rob's paper, and we'll have this demo data set available on all of the Cytobank servers with Citrus enabled, which is, at all enterprise and our premium servers. So you can try this out yourself if you'd like to, if you have an active Cytobank account. We'll have this data set up there. So the model with the lowest estimated error rate has phospho-S6 as the single strongest predictor of whether a file should go for one group or the other. And this is on the B cells. So if we see this this model error, or basically the feature plots version, it's saying this node in phospho-S6 is the best predictor of whether a file should go in the unstimulated or the stimulated group. The FDR constrained version of the results, however, has not just the single strongest predictor, but all of the differences between the two groups. So this could be, like in this case, it was stimulated with B cell receptor cross-linking, but it's showing that the phospho-AKT on the helper T cells and on this myeloid branch is changing as well. So there may be off-target effects, and this is what makes this FDR constrained model a good hypothesis generating tool. It lets you kind of explore all the differences between these two groups. Again, another PDF you'll get is the phenotype of any significant cluster. So the CV-min, in this case, just has one set of histograms that describe the phenotype of this node in particular. And we look at it, it says CV20, HLA-DR, IgM, that's a dead giveaway for a B cell. FDR constrained version has many, again, significant nodes and many phenotypes associated with them. Last PDF you'll get is a box plot showing you for this cluster, which is the B cells, and the phospho-S6, this is where those values are, the transform values sit. So you can see there's 8 dots here for the reference samples and 8 dots here for the BCR stimulated samples, and that is quite a large difference. Lastly, you'll get a CSV file of these values that are plotted on this box plot. So if you would like further reading, pardon me, on Citrus instead of just on the debut paper, we have here the reference for this paper that Gabby Fragidakis will be talking about in just a little bit. Again, this is a great paper. Check it out if you'd like to see how Citrus works kind of in the real world. So now briefly, some new developments in Cytobank. We talked about Citrus. We also have gate tailoring across populations. So this is a kind of a more traditional method for looking at gates on functional endpoints. You don't have to draw 5 gates now on your phospho markers. You can just draw one and tailor it to its parent population. And then lastly, we have a Cytobank API that's been recently released. And this is more for computational biologists and bioinformaticians, but it's a way to computationally interact with Cytobank. So instead of having to click around in the user interface, you can send Cytobank a script or a list of commands in R, and it will do the work for you. With that, if you have any questions after this webinar or in the future as you get started with Citrus, please go to support.cytobank.org and write in. We'd love to hear from you either for feature requests or also from feedback if you need help with some of these. So with that, we'll transition over. I'll do a brief introduction, and thank you very much. So It's my pleasure to introduce our guest speaker, Dr. Gabriela Fragidakis. Dr. Fragidakis' graduate work applied mass cytometry to monitor the immune system both in surgery patients and in healthy individuals. And she earned her PhD in microbiology and immunology from Stanford. Today, her postdoctoral work with Dr. Justin Sonnenberg at Stanford explores integrating immune monitoring and microbiota profiling. She will now discuss how applying citrus led to clinical insights in her 2014 Science Translational Medicine paper, Clinical Recovery from Surgery Correlates with Single-Cell Immune Signatures. With that, you have the floor. I'll pass this presenter back.
Sound check test. Great. Well, hi everyone. Thanks, Jeff, for that great introduction. As I said, my name is Gabby Fragidakis, and I'd like to talk to you about work that I did during my PhD relating early immune responses to recovery time in surgery patients. And in doing so, I hope that I can illustrate the utility of citrus and the citrus package in data analysis, because this tool was very much integral to finding our results in this work. So I'd like to start by mentioning that this work was done in full partnership with Dr. Brice Gaudier, who at the time was a clinical fellow and postdoc in Dr. Gary Nolan's lab with me, and is now running his own lab at Stanford. And so this work really started with this question that we had of how at the beginning of surgery, or pre-surgery rather, there can be two patients who, for all intents and purposes, look identical clinically in the ways that we profile and evaluate patient health. They're indistinguishable. But then over the course of the surgery, one patient can, go through it fine and recover, over the course of a week or two, whereas another patient could have an adverse response and, be in bed with complications for months. And, this relates to something that many of you may have experienced if you've gone any form of surgery where a surgeon tells you, your recovery time will have some long range, 2 to 8 weeks, without any sort of indication of what end of the spectrum you'll fall into. So we really don't know this information ahead of time of how the surgery is going to go. However, we do know that a successful surgical response is dependent on a successful immune response. So how the body responds to surgical intervention is have a wave of inflammation immediately following the surgery to respond to the wound, largely governed by your innate immune cells. But then that inflammation is reined in and tempered in a wave of immunosuppression, largely governed by your adaptive immune cells, in order to sort of mitigate damage from the inflammatory response itself. And any dysregulation on either end of this, either uncontrolled inflammation or too much immunosuppression, can lead to complications and outcomes. And so we had this question of, because a successful immune response, successful surgical response depends so much on a successful immune response, could we actually track patient-specific immune responses in order to predict their recovery time? And so to do this, we decided to look at patients at Stanford Hospital undergoing hip replacement surgery. So we chose hip replacement surgery since it's a major surgery, but patients immunologically are relatively healthy. And we, sort of monitored their demographics and their vitals, and as well as their recovery time in ways that I'll talk about shortly. But what we did is we took blood samples from these patients at their preoperative baseline and several time points after the surgery, including 1 hour after, 24 hours after, 72 hours after, and 1 month out. We critically barcoded those blood samples together. We stained them with antibodies that would allow us to identify different immune cell populations, as well as look at functional attributes, so in this case, phosphosignaling. We then analyzed them by mass cytometry, or CyTOF, and critically normalized that signal, debarcoded the signal, and performed the analysis that I'll talk about. And so along with this, sort of along with our profiling of the immune responses, we of course were tracking recovery time. So before I get into the full data that we profiled for recovery time, I just want to show that we were sort of indeed seeing this pattern of kind of this wide range of recovery time. So here I'm just going to show you 3 patients on one of our scales of recovery, which is the SRS scale, or the surgical recovery scale. And here in this case, 100 is high functioning, it's feeling perfect, and 0, you're in trouble. And we're just going to look at the time course of 3 patients in the days following the surgery. So here, we had one patient who, had the initial drop, and then after a few weeks gets to their level of sort of their preoperative functioning score. But then, we had another patient who also had that initial drop, but really struggled to get up to that point of their preoperative baseline. And then we also had patients who also had that drop, but then, really after a week are doing better than ever, sort of in this remarkable way. So we were really seeing this range that had been reported of different sort of trajectories of recovery. And so we were able to track this trajectory for all 26 of our patients in this study on 3 metrics: the surgical recovery scale, as I mentioned, a measure of hip function. Remember that these are hip replacement patients, and also how much pain they're in. And what we can see is, while ultimately most patients do recover, you definitely see this sort of wide range in the recovery time. And so really our question was, could we predict from early immune responses that we were measuring by CyTOF sort of what camp you're going to fall into, how long it was going to take for you to recover on these 3 metrics of clinical recovery? So to do this, and actually I want to give the caveat that I'm going to talk about the citrus analysis that we did, but there's also many other analyses that were useful in this project to really investigate and explore the data, many of which we did using Cytobank, the gating functionality, other sorts of manual exploration of the data, which is very important. But I'm going to sort of zero in on this one part. So that is to say, something that we always would say in the Nolan lab is to manage expectations in terms of what one of these experiments looks like. If you're doing a major CyTOF experiment, you're probably spending a long time optimizing your reagents, baking your samples properly, et cetera. And then you run the CyTOF experiment, but sort of just as much time needs to be spent on the data analysis, and to do so in a very thoughtful, iterative way. And a big component of that for us was the citrus analysis. And so really the challenge becomes, from your CyTOF run, you basically get a file that looks like this. You get an SDS file that's essentially a matrix where you have these different cell events. And for each one of the cell events that was profiled by CyTOF, you have a measurement of, say 40 or 50 proteins, so antibodies corresponding to specific proteins that you've designed. But really, you don't just have one of these. So as Jeff mentioned, we're looking at groups of samples, groups of patients, and so really you end up having many of these matrices that you're trying to make sense of. And perhaps maybe you also have some measure of an endpoint or some information that corresponds to the patient that you're interested in. So, this could be 2 groups like, as Jeff mentioned, a responder or non-responder to a cancer treatment. That could be A and B. In our case, we have a continuous variable of recovery time. So we have information about each of these patients along with their CyTOF measurements. And so what would be nice is basically to get our CyTOF data in such a form where we have, for each one of these patients, we have sort of associated features and feature values for them, and then that we could build a model that relates those associated sets of features to our clinical endpoint of interest. And really what Citrus does is kind of take us through this whole process that I'm showing here. And so to kind of bring it back to what Jeff explained, CITRUS is this method for identifying cell subsets, calculating properties of those subsets on a per sample basis, creating models of these features with an experimental endpoint, and then actually building the model in such a way where you can go back and see what were my informative features, what was the biology that allowed us to predict our clinical endpoint. And so I'll go through each of these steps and then be tying it into how we did it specifically for the surgery data. So first, with identifying our subsets, as Jeff said, we do hierarchical clustering. And so as we're sort of breaking down these cells into groups, this is done based on the expression of cell surface markers typically. And why this is important is, you can think of this as analogous to manual gating when we're sort of defining populations based on cell surface marker expression, but this is done in a relatively unsupervised manner. So just as opposed to having the user go in and draw boxes in around the populations that they know and love, this is done more systematically with hierarchical clustering. And importantly, in your analysis, you're actually you'll set some sort of cutoff point for the hierarchical clustering, but then each one of these populations going down from all cells until your cutoff point are going to be used in the analysis. And why that's important is we don't really know sort of at what level of resolution your data is going to be the most informative. And so basically, as was shown with Jeff, you'll get a clustering hierarchy that you'll then be able to investigate based on the expression of the surface markers. And so this gives us our populations. You can think of this as like our gated populations, as I said. So we have this set of populations, and then with that, we want to have an associated set of feature values that will be for each patient. So in this case, we did measure signaling responses, and so for each one of these clusters, we can have the levels of PLC gamma 2, ERK, P38, et cetera, for each one of our patients. So basically this ends up looking like this, where you have each of our patients in our study, and there'll be an associated level of per cluster per protein, and those will make up the feature set that will then be used to build your model. And so next, like I said, we want to incorporate the clinical endpoint and basically build a model to relate the two. And when we did this, we were actually able to find relationships from our 3 different metrics of clinical recovery. So here I'm showing 3 plots. They each correspond, as you can see in the title of each plot, to one of our metrics of clinical recovery. And here we see significant correlations between the length of recovery time and functional responses in 3 different clusters here that I'm just calling cluster A, cluster B, and cluster C. So basically what we can see is we're able to, in this case, I'm showing the results of SAM, which is one of the models that is included in CITRUS that Jeff talked about. Here we see that we can find a relationship between these early immune responses to surgery and recovery time, and CITRUS allowed us to do that. Let's see. And so what we wanted to make sure of, though, is, critically what we care about as biologists is, it's great that we are able to find something predictive, but we want to know the biology. We want to know, what are cluster A, cluster B, and cluster C, what allowed us to predict that, because then maybe we, wouldn't need to do such an extensive profiling in the future if we wanted to, for example, develop a diagnostic test. So basically, the last step in CITRUS that it was very important to this work is this idea of when we go and we have our feature set, figuring out what features allowed us to predict our clinical endpoint, and then what is the biology of those features. And so when we looked in our case, if we go back to our hierarchy, we found that here I'm just highlighting in red the clusters that were predictive for us, and when we go and delve into what those clusters actually are, we found that they were populations of monocytes, particularly populations of HLA-GR low monocytes. And so this now told us that sort of the potent indicators that we had were the signaling responses in the specific monocyte subsets, and those were something that then we could go and follow up on and understand why biologically those were such potent indicators. And so to sum this up, Citrus was incredibly important in connecting our Cytobank data to our clinical endpoints. It takes us from that raw data to building interpretable models of clinical endpoints, interpretable being really important here for us to get at the biology. And the biology that we ended up finding here was that signaling responses in particular monocyte subsets were predictive of recovery from surgery in patients. And with that, I do want to thank other members in the lab that were critical for doing this work. Of course, Rob Brugner, who we worked closely with as he was developing the Citrus algorithm, and sort of throwing many use cases at him for thinking. Dr. Gary Nolan, other members of the lab, Dr. Brice Godier, who I said we did this in full collaboration, and our clinical team, Martin Inks lab, for enabling us to do a clinical project. And that is thank you very much for listening.
Great. Thanks so much, Gabby, for that. Now, actually, we're going to hop into a brief demo of how Citrus looks in the software. We'll take a little bit using Rob's data set that they published that first paper on, and show how to set up a Citrus in Cytobank, and also what the results look like. And then once we do that, we'll hop into our Q&A session. So we'll just take a few minutes, and I'll show you, like I said, what it looks like in the software. All right, so once you sign into Cytobank and upload your data, again, ideally at least 8 samples per group. So in this case, there's 2 groups, 8 unstimulated and 8 BCR cross-linked samples. Then you can start on Citrus. Just like Visney or Spade, it's best if you clean up the data a little bit beforehand, both looking at singlets, live cells, potentially CD45 if you're looking at immune system stuff. And from there, you can start off a Citrus run. So up at the top here, we'll click on Citrus, New Citrus Analysis. We can give it a name. And we're presented with the setup page. And again, I said it's a little more complicated than setting up Visney or Spade. And this is all right. We'll walk you through it and we'll take some time. So under population, this is similar to our other algorithms. You click on your kind of nice, clean beginning population, in this case CD45. Under clustering channels, we can do CD markers. And we'll click all. It's also, if you have non-CD markers like IgM, still important, HLA-DR, still important, include those as well. It's important to note here that if you're planning on doing a median cluster characterization, looking at a functional endpoint, to not include those functional markers in the clustering channels. So simplest, again, is just to use surface markers for clustering, and then if you're going to look at medians, save those functional markers for that. And then lastly, under file grouping, here we have to indicate to the software which files are in which groups. If you name your FCS files, just like other places in Cytobank, if you name your FCS files something relevant or to how they were treated, Cytobank will sort it out for you. So in this case, if we call something reference and BCRXL, click OK, Cytobank will match them, the file name, to the group and automatically sort them out for you. If it doesn't do that, if you haven't named your FCS files things, you can always manually change which group it belongs to. So we'll click close. So this is the first setup portion. Again, like other algorithms, it's important if you're using fluorescent data to include a very nice clean compensation. This is not, so we don't have to worry about that right now. Here, because we have only 2 groups, we can run all 3 association models if we want to. However, I'll be just debuting this PAM version. Under medians or abundance, we'll select medians and then choose all of the phosphosignaling markers. Again, there's a search bar up here where you can go through and look for relevant markers. In this case, we'll use all the phosphosignaling ones. We'll click close. Event sampling, here you can either choose to pull an equal number of events from your files or the maximum per file. We'll choose equal in this case. And then you have some additional settings here. If we do equal, you can see how many events will be pulled from each file. The minimum cluster size, we're looking for B cells here, so 5% is a good minimum cluster size. However, you can change this cluster size if you're looking for rarer events. This is essentially saying how large you want those final endpoint clusters to be on that tree. You can lower this if you're looking for rarer cells. Cross-validation fold changes, 5 is a good start. But again, this is user variable, and we have lots of documentation up online about the best options for each of these. And then the false discovery rate, we've set a default as %. From here, that's all we have to do. That's all we have to set up. We've chosen how we want to sample, what medians we want to look for, and what starting population we need. And then we'll click Run Citrus Analysis. However, we're not going to sit here and wait for this analysis to complete. We'll pull a little Martha Stewart, actually, and here is a completed citrus run. This is what it looks like. So you can see the settings we used to set up this population or this run, and we'll get to this in just a second. From here, actually, we can download the results. I've already downloaded them, so we can see these results in our download folder. There are some supporting files, things like the R analysis logs and the results that people are looking for. The marker plots, again, this is a PDF of the phenotype of all of these different citrus runs. And then the PAM results, we have a series of PDFs, some CSV files, and then down here is the model error rate graph. So you can see here, after the first number of features, it has now approached 0, this cross-validation error rate, and that's where the CV-min and CV1SE are. So this, because both of these are off of this y-axis, cross-validation error rate approaches 0, and the feature false discovery rate doesn't increase until later. I'd say this has a very good chance of being a solid run and good results. So we'll take a look at these other PDFs real quick. So the feature plots for the CV1SC, you can see that this node right here is the one with the strongest predictive value between which group the file should go into for phospho-S6. And if we go back and check this out, this is the B cells. So let's take a look at the clusters. So here is the cluster information for that: CD20 high, CD7 negative, IgM, HLA-DR positive. So yes, those are our B cells. If we want to look at the box plots, we can see that box plot there with those values. And here is the CSV file with those values in place. So this is the result of a citrus run, which is all fine and good. You say, that's fantastic. Now I know which node has my cells of interest in them. But what do I do now? So instead of just tantalizing you and saying, we have your results here, but you can't do anything with it, we give you a way to explore those results more. So using this button, Export Clusters to New Experiment, you can see which clusters are interesting. And if we go and take a look back here, feature plots, it's 34067. That's the node number. Again, the size of the node approximates how many events are in that node. That number is just for identification purposes. I've gone and forgotten what it is, 34067. And we can hop in here and say, all right, this node, I want to take my B cells into a new Cytobank experiment. So it'll give you the events from that node from each of your files in a new Cytobank experiment. You can select more than one node if you'd like to, and you can include original files if that'd be your preference. You click Start Export, and they'll give you a new Cytobank experiment with basically a curated number of events from this original citrus analysis to then do further analysis on. So citrus is a great tool for discovery, but it's always, like Abby said, it's always important to go through and confirm your results using a traditional analysis. So that's a brief run through of how citrus works in Cytobank. We'll take it now, and we'll hand the floor over to Rob. So I'll do an intro quick. We're very fortunate to have citrus creator, Dr. Robert Brugner, joining us today for the live Q&A portion of our webinar. During his graduate studies, Rob investigated high-dimensional single-cell proteomics data with a focus on identification of cellular-level signatures of disease and other phenotypes, and earned his PhD in biomedical informatics from Stanford. Today, Rob uses his computational expertise as Director of Data Science at Nuna, a healthcare analytics startup in San Francisco that helps companies understand health insurance benefits. We'll kick off the live Q&A session with Rob, sharing why he created Citrus and how he sees the computational tools advancing our understanding of human health today. So, Rob, take the floor.
Hi there, everybody. Great to have the opportunity to speak with you all today. And I have to say, watching the ways people apply Citrus in new and interesting ways is actually probably the most rewarding thing about having done graduate research in the first place. So I'm excited to see what people learn in the space and how they apply things. To Jeff's question originally about why did I create Citrus and why do that? As an incoming graduate student, I was really excited to learn about the power of single-cell experiments, particularly with respect to identifying these cellular subpopulations that could act as strong biomarkers for disease progression or clinical outcomes, drug response, things like that. And I distinctly remember a paper by Yael Garnes in the Herzenberg lab that showed just how you could mix blood from a person with some peanuts in a little test tube and then measure basophil activation using flow cytometry, and you could precisely see whether this person was allergic to peanuts just by doing this little in vitro experiment. And so I thought this was incredibly powerful. And I thought that this challenge of identifying informative biomarker populations was a really useful area in which to conduct research, particularly with respect to the applied clinical setting. And since my graduate program was actually in biomedical informatics, I thought that it would be both a useful and interesting thing to work on to see if I could actually enable this process of discovering these cellular biomarker populations using efficient and automated computational methods. And so I began to dig into this problem. How could I efficiently enable people to identify these cellular biomarkers? And so as I dug in, I learned about more about the task of why identifying these populations is hard, and what are at least a couple of major blocking issues became very apparent to me. First, there were real concrete limits on the number of parameters that you could simultaneously measure in any given cell and experiment, and that means that if you were trying to discover a new biomarker population, you were limited to the number of cellular subsets that you could measure in any given experimental run. And what if the informative signal, what if that population is actually not detectable using that panel of, say, 8 to 10 markers that you selected for your experiment? So that seems to be a pretty big challenge. The second blocker that I noticed was the process of identifying populations to inspect for biomarker potential is manual gating. And manual gating is incredibly powerful in that it makes use of a decade of immunological knowledge. But what if that informative population that you're looking for is one that we don't really know exists? So how would we find it? So those were two big challenges that I was trying to understand how to overcome to enable biomarker discovery. With respect to that first challenge in terms of limitation on biomarkers, about the time that I started my graduate work and joined Gary Nolan's lab, we started working with one of the first mass cytometers to perform actual biological and clinical research. And this machine could measure 40, 50 parameters at the same time, and it quickly became clear that the data that this machine was capable of measuring we could collect far more markers and detect far more populations than we could ever hope to evaluate using standard, manual gating techniques. So that kind of took care of that first problem of measuring a limited number of populations. And with respect to that second challenge of basically, you know, how do we sort of dig through the data and find the informative populations, even if we don't know that, you know, these populations existed, it was actually very clear to me, well, sorry, it seemed very possible that these new applications and computational algorithms that are sort of being developed in the informatics space would be a great way to actually automatically identify these informative populations in this highly complex data, and specifically, you know, take care of the process of first finding those populations, and then second, telling, you know, and automatically determining which ones of those were associated with some clinical endpoints, and, you know, things like drug response, phenotypes, clinical outcomes, et cetera. So, you know, the methods and the confluence of the new data really seemed, you know, ripe for the creation of things like Citrus. So that's effectively where the motivation came from in sort of 2 minutes or less. And the conclusion to this story is that I spent the next 3 years of graduate school trying to learn what the best computational and statistical tools were for first identifying cellular subpopulations automatically, and second, determining which were actually associated with your clinical outcomes of interest. And all of that research ended up coming together in the Citrus package that you've seen demonstrated here today. So that really speaks to the origin story of Citrus and what the motivation was effectively to create this. With respect to Jeff's second question, which was, you know, sort of how do I see computational tools helping advance our understanding of human health today? The things that I'm really most excited about, I guess, in the space, speak to the maturation of data-driven techniques for biological or performing biological research, and that is, I'd like to draw, you know, sort of a comparison between the existing hypothesis-driven research that is a core and fundamental principle of science today, and it's been used, you know, for the last, you know, couple hundred years to advance the state of science in the world, versus these new data-driven techniques that are sort of becoming available to us today, which is, you know, wherein we gather tons and tons of data and, you know, dump it into these computational algorithms and out emerge patterns, things that we, you know, never anticipated seeing. And, you know, the possibilities that sort of allow us to open up our mind and explore the possibility of new things that we just don't expect to be true, and have those things revealed to us without any sort of prior biases towards what is or is not possible. I think the things that are necessary to support that really are, you know, sort of these large-scale data collection efforts, these biobanking efforts that effectively are collecting and capturing, you know, large data sets, and in the, you know, sort of the long-term clinical, you know, outcomes of the individuals associated, you know, with these with these samples. And I strongly believe that, you know, at the end of the day, that we are going to be able to put these data together, take these sort of long-term perspectives, and determine effects that we never could have seen using sort of small scale or limited scale data. This is not to say that hypothesis-driven research is not you know, useful or informative. It absolutely is. I think that the data-driven you know, techniques will just become a very complementary approach to performing research, you know, as we see it today, and just give us more options when it comes to presenting hypotheses and paths to explore, et cetera, et cetera. So those are the things that I'm particularly excited about with respect to computational immunology.
Excellent. Thanks so much for sharing. Now we're going to hop into a few questions from the audience. So the first one is for you, Rob. It says citrus clusters look a lot like spade trees. Does citrus clustering work like spade?
Short answer, yes and no. It uses the same fundamental clustering algorithm that is just standard agglomerative hierarchical clustering. The major difference is that citrus actually identifies a hierarchy of related clusters. That is to say, it might identify, you know, one cluster of B cells and then partition those B cells into IgM positive and IgM negative B cells, and it feeds both that population, that superset of cells, that entire B cell population, and the respective subpopulations, the IgM negative and the IgM positive, B cells into the association detection algorithms. And so there's redundancy in these clusters. So that was a very specific and intentional design decision for the algorithm, because we expect that the association algorithms will actually tell us which of these subsets is actually the most informative. And so that is the principal difference between Citrus's clustering and Spade's. Spade, in fact, separates everything out into separate, mutually exclusive subpopulations, and connects them all with a minimum spanning tree, where Citrus identifies a bunch of related and redundant clusters and lets the supervised learning algorithm sort out which ones are informative with respect to your clinical endpoint.
Great. Another one for Rob. Are the clusters or nodes Citrus produces the same in all of the sample groups? Do you expect the clusters to have different or unique features? Related, if a population of cells is only in one sample group, will Citrus still pick up that in its clustering strategy?
Yeah, so the first part is, do the clusters look the same, and will they have different properties? All of the clusters, the subpopulations, are identified by bringing and collapsing all of the individual events from each of your different samples together. And when you perform a clustering that is based on the phenotypic markers, the cellular surface markers of the cells, that means all the different cells from each of the different samples will get clustered together. And so, from a phenotypic perspective, this means yes, all of the cells actually, or all of the clusters that are identified from all of the different samples should look phenotypically similar with respect to the clustering. The second part is whether or not they look identical from all of their properties, and I think the answer to that should be no. So the discrete properties that you might evaluate of those subpopulations might be things like what proportion of this sample cells are actually IgM positive B cells, and that we expect to vary between samples. Or maybe you want to say what is the median level of this phospho S6 marker in our B cells for this sample or for all of our other samples. We expect those features to be different. So between our samples. So the answer is yes and no. They should look similar from a phenotypic perspective, but from the properties that we evaluate with respect to cellular abundance or functional activity, we would say no. We do not expect them to look similar. Jeff, sorry, what was the second part? What was the second question that you asked? If a population of cells is only in one sample group, will Citrus pick that up in its clustering strategy? Oh, yes. Because all the cells are brought together during the clustering step, we would expect that those cells would be part of the overall clustering set of clusters that are identified across all the samples. Okay, great. Another one. How many samples are necessary to get meaningful data in Citrus? Do comparison groups need to be balanced? The famous statistical power question. My first and foremost recommendation is that you make friends with statisticians at your respective institutions, because it's always good to be friends with statisticians because they will help you interpret your data. With respect to performing comparisons between Citrus, I definitely would recommend using at least 8 samples per group, more if you can have it. And the intuition behind this is simply, let's say I'm doing a study about, I'm trying to understand phenotypic effects of characteristics of AML-like cells in cancer. If you wanted to say something about the general population of patients with AML, you wouldn't just look at, say, two patients and look at their blood and say, okay, now I understand what AML looks like. You know, the more samples you use, and if you look across, say, 10 or 20 or 30 different individuals, and you see trends across those 10, 20, or 30 individuals, then you have more confidence that what you're seeing is actually representative of the population. So I would definitely recommend using at least 8 to 10, especially, and more if you want to try and make a general characterization about all individuals with this phenotypic condition. The second question is do the sample groups need to be balanced between citrus? The answer is no, they do not, although if the sample groups are unbalanced, you just need to keep in mind that the performance of the model is going to be, at worst, you know, the performance of your predictive model is really going to be influenced by the unbalanced nature of your group. That is to say, if I have 100 samples and 90 of them are from patients with no disease and 10 of them are from patients with a particular disease, and I can create a predictive model that's 90% accurate, that's not actually that good, because I would by default guess with no other information that you know, nobody has the disease. So when interpreting the results, you just need to take that into account in evaluating the performance of the predictive model.
Okay, and one more. Does Citrus handle correcting for multiple hypothesis testing?
It's different depending on the model that you select. For the PAM and LASSO regression models, it's not actually performing formal hypothesis testing in the classical t-test, you know, sense of the word. It's really performing a regression, and so we use standard evaluation and classification of techniques to evaluate the sort of the false positive rates, the accuracy, the sensitivity, and the specificity of the models, respectively. For the SAM, the associative model, that actually controls for the overall false discovery rate of the model instead of explicitly doing a sort of like a statistical correction for multiple hypothesis testing. So the answer is yes, it does in fact control both, you know, for multiple hypothesis testing, although each of the different models does it in a different way.
Okay, great. And with that, actually, we're going to wrap it up. For those of you whose questions we didn't get to, thank you so much for writing in, and we will respond via email. If you have any questions after taking a look at Citrus for the first time, or any further comments on the algorithm, please write in. Go to support.cytobank.org and write us an email. And also, we've recorded this webinar. We'll be sending out the link in the kind of a thank you follow-up email. So please feel free to watch it again and share it with anyone in your groups who missed it. And with that, we'd like to say thank you very much for your time today, and have a good day.